All right, everybody. This is not exactly the way I wanted to do notes. I would rather let you see my shining face and the little screen in the corner, but it's not going to work out that way. We're having some difficulties, and I apologize for that. I don't know where I was in all the different classes, so I'm going to run through the last little bit because we've talked about several things. First off, Mendel and his peas. 5 main reasons that he used the peas in his monastery up in Brunn, Austria. Peas are very easy to find pure breeding strains. If you want all tall peas, they will be all tall peas. If you want all wrinkled seeds, they'll be all wrinkled seeds. Peas can self-fertilize. So if you have one pea plant with flowers, you have enough. Peas can be easily grown in a greenhouse. Don't forget, we're in Austria. It's winter. You're still going to need food. So you can do that in a greenhouse. They produce a lot of offspring in a short amount of time. You figure every pod has 6, 7, 8, maybe even 9 different peas in it. And they stick to the probability ratios very nicely. Now, Mendel didn't understand this. Okay, he didn't understand why it stuck to the probability ratios nicely. That wasn't discovered until the 1920s when they found his work and started to analyze it. So, what he didn't really understand was genes. Genes are a location on the chromosome, as it says right here, and I know the screen keeps going different. It's a location on the strand of DNA that's responsible for passing on the traits. For example, some of you have the trait for tasting PTC. We all have the trait. It's whether or not it is turned on or turned off. And that's what's responsible for passing on your traits. Mendel didn't know any of this. It's 100 years before DNA is even discovered in 1953. Okay, more than 100 years actually. Okay, so here's a real quick look at how this all occurs. Of course, we've seen the cell earlier this year. We know that the cell at the very, very center has the nucleus, or somewhere located near the center. We know that in that nucleus there is DNA, and that's what you see over here. Okay, DNA is a double helix, and we'll be learning more about that in a couple of videos from now. And it's wrapped around these small balls called histones, and it supercoils into chromosomes. Okay, and the chromosomes, of course, like I said, are kind of like a loaf of bread. Each slice of the bread has the potential to be a different gene. Okay, we're going to move on. Next page, alleles. Excuse me, once again. Pairs of genes that carry the same trait and are located in the same place on the pair of chromosomes. So when your birth mother and birth father exchanged chromosomes upon fertilization, the dominant gene or the recessive gene was in the same exact place on mom's chromosome number 3, let's say, and dad's chromosome number 3. And when those chromosomes align and start to wrap around each other and exchange information, that's when the dominant gene is actually pushed in or expressed in the only way, of course, that you can get the recessive is with 2 recessive genes. Okay, so we already explained what chromosomes are, the supercoils. I am going to take some stuff home and do a video of so you can see what I mean by supercoiled. Okay, the locus is the particular spot. Okay, just like a map has little dots that show where a city or a village is. That's what the locus is. Okay, the locus is a specific spot on a chromosome where that trait can be found. Okay. And we've already kind of briefly mentioned what a homologous chromosome is, one from mom, one from dad, birth mother and birth father once again. So the alleles are always in the same position on those homologous chromosomes. So it's very much like if you reach your hand out and someone else reaches their hand out, you can grab hands. Of course, you shouldn't be doing that right now, but they are in the same spot. Okay. Some more of those terms that we started to talk about, and we've used in class and lab, homozygous and heterozygous. Remember, homozygous is same. Heterozygous is different letters. Okay, so homozygous might be something like big T, or little t, or big S, little s. It doesn't make it any better. It doesn't make it any worse. Okay, same as dominant and recessive. Dominant is no better than recessive. It's just different. That's all. There's this negative connotation about recessive genes. Okay, parental. Your parental generation is the people that gave birth to you. Okay, your birth mother and your birth father. I don't believe any of you are P1s yet. You are not parents, but you are all F1s. Your F1s of your parents, your F2s of your grandparents, your F3s of your great-grandparents, and it keeps going back like that if you get into genealogy. Okay, So here's that F2, okay? You are the F2 of your grandparents. Their F1s, your grandparents' F1s, are your birth parents, okay? Mendel's main laws, once again. First, the law of dominance. And it says if the dominant trait is expressed, or if the dominant trait is present, it will be expressed, okay? That trait will most often be expressed. Dominant, of course, is some people call it the stronger gene, and that's not really a good representation of it. It is just one that will be expressed more often than the recessive. The only way you're going to get the recessive is if you have two recessives, one from mom and one from dad. Moving on a little bit again. Progeny, your offspring. You do not have any progeny yet, okay? They will genetically be about 50% of each of you, both mother and father. Okay, so when you have children, people will say, oh, he's got his father's eyes, or he's got his mother's hair. That's pretty accurate, actually. Okay, siblings, these are your biological brothers and sisters. Okay, they do not have to share the same biological parents. They have to share one parent. Okay, so if there's a family situation where mom or dad has a new spouse and they have a child, technically if they have that child, it is a partial sibling to you. Punnett squares, you got to know and love them, you know that. Okay, those are the prediction method that we talked about, that Reginald Crundle Punnett came up with, and I think you have that in your notes already. Phenotype, phenophysical features, phenophysical features, phenophysical features. I can't say that enough. Phenotype will be what you actually see when you look at someone. Okay. Law of segregation or recombination. During sexual reproduction, which of course you are the product of, the traits segregate and when the gametes are made and they randomly recombine. That's why it's pretty much impossible to have any two individuals on the planet, well, not even pretty much, it is impossible for any two individuals on the planet to have the same exact genetic makeup. If you commit a crime and they find your DNA, it's pretty much lockstep, 100% assured that it was you. Okay. Law of independent assortment. Excuse me, once again, if the genes for two different traits are located on different chromosomes, non-homologous, they may not be inherited together. So it's very common that people associate different genetic traits with each other. Oh, if he's blonde-haired and blue-eyed, mainly like people that we usually refer to, people in Sweden, that's usually a characteristic that we see. Oh, they're blonde-haired and blue-eyed, and they're these gorgeous people. You could very easily have a green-haired, green-haired. No, let's not go with green hair. Let's go with green eyes and brown-haired person in Sweden with two parents with blonde hair and blue eyes. Now, the blonde hair and blue eyes is more likely, but you can get those differences as well. Okay. Intermediate inheritance. I love intermediate inheritance. I love it for a couple of reasons. One, because I got in trouble with it one time with my mother's tulips. We'll save that story for another day. Okay, and also because it's really cool to see how your blood types occur. Now, you've had a blood type lab earlier this year where we looked at the percentage of people that have AB, ABNO, Rh positive, Rh negative blood. You drew a graph. You figured out how much blood was yours. The reason that you have your blood type, and it might be different from your parents, is because of codominance or incomplete dominance, okay? Now, I know all along I've said you either have a dominant gene or a recessive. Well, I'm going to make the rule change a little bit here. It is possible to have two dominant genes, okay, two alleles that are both dominant. Okay. Other examples, there's some horses called roans. There's a chicken called Andalusians, and they actually do look blue or gray, okay, like a slate gray. And then other things in the human blood types like sickle cell anemia and that sort of thing. So this graph right here kind of demonstrates codominance, and I am going to somehow, somewhere, some way magically produce a lab that will go through and discuss this a little bit more. The real fun of this, honestly, is to do the actual Punnett squares. and I'll have a worksheet ready for you. Actually, I kind of have it almost done, and I'll post that so you can try it. It's actually really cool. Okay, I know genetics for many of you is not what I would term really cool, but I really enjoy it. And I'll put up a separate video on codominance and blood types and that sort of thing. Also, whether or not you have good or bad cholesterol. If you have a cholesterol problem in your family, that is also a incomplete dominance trait. Okay. Oh, here, well, hey, here it is. I forgot I had it in here. Okay. You can have 3 different genotypes for what's called hypercholesterolemia. Okay. If you have high cholesterol. Okay. If you are normal, okay, you have these receptors on the outside of the cell, and don't forget that's how signals get into the cell. Okay. If you are a normal person, you will be big H, big H. It means you're homozygous for the ability to make these receptors right here that I'm kind of highlighting. That way, when the LDL comes in, it stands for low density lipoprotein. Don't worry about that. Okay. It can hook on and bring the LDL, the fat, if you will, into the cell and use it for energy, use it for processing. If you're heterozygous, you have mild high cholesterol. You don't have as many of these receptors on the outside. So a lot of this cholesterol stays in your bloodstream. Don't forget, this is a cell. You could have blood zooming through here right next to it. Okay, if you're heterozygous, big H, little h, you have a few of those receptors, but you don't have a lot. So your cholesterol, your blood cholesterol, once again, might be a little bit higher. If you are homozygous recessive, little h, you have the inability to make those LDL receptors, so you suffer severe disease. All that cholesterol that you take in stays in your bloodstream. It doesn't get taken into the cells and used for energy and that sort of thing. And that is a concern. People with this inability to make these LDL receptors will have a higher blood cholesterol, therefore they have a higher chance of cardiovascular disease as well. Okay. Let's see what else we got. I think we're on to the next part here. And I'm not going to go into too much. Actually, in your notes, you have these multiple choice questions. These would be very good to look through and see if you can figure it out. Most or all of these actually are pretty much just a standard Punnett square that I have confidence that you could figure out. We've already covered monohybrid and dihybrid crosses. This here is just a Regents question. I know it has nothing to do with what we're talking about, but it's just a little bit of constructed and extended response review. Next, we're going to get into genes and mutation and anomalies. I'll put that video up in probably the next 48 hours or so. But until then, thank you very much. As I mentioned earlier, pause the video, take the notes, watch the video. I gave instructions on Google for that. Take care. We'll see you soon. Bye-bye.